this seminar. We've been having joint lab meetings to talk about this, and I want to acknowledge the lab members. I won't go through all the names, but there are numerous, there's undergraduate students, master's students, PhD students, and postdocs who have been helping with this. And so today it's really exciting to start this. The idea of this find is that we want to engage everyone in discussions about social evolution. And we wanted to bring together a diverse group of people. I think we've done that. It's really exciting if you look at the number of people, the diversity of talks that we're getting we have people coming from various countries giving talks. And this really gives an opportunity for, I think, people who are, obviously we can't travel now, but even those who normally can't travel, to engage in discussion about this exciting topic. In terms of the types of talks you're going to be hearing through December, we have a diverse talks ranging from ants to spiders to birds, and of course there's fish talks and then mammal talks as well. The structure of the seminar, we hope, is the three co-hosts, Eduardo, Carsten, and myself, will lead off giving talks so that we can work out some of these technical difficulties and anticipate some that those of you who give talks in the future hopefully won't have to deal with. The participant list, as we said, it's really wonderful. We've had a lot of people reaching out and interested. We have over 200 people on our email list from almost 20 countries. And of course, there's lots of students and postdocs, which we're really glad to see. We aim for growth. So if you know people in the field that want to participate in this, please have them contact one of us by email, and we'll add them to the email list, and hopefully they can participate. Just as you know, many of you probably know already, we're also live streaming this to YouTube, and that will be recorded. So those of you who can attend on a given Tuesday, hopefully you can go and check it out later. We just have a couple of rules. This is all new to everyone, I think. And so we're going to ask for just a few minor rules. I think we've said it that the chat function is exclusive to the Q&A. We ask that you don't type anything in the chat until the Q&A session, just so that I think it's set now that only Carsten and I can see it, but the hope is that that won't distract Carsten during his talk. We also ask that you please keep the microphones on mute so that there's no background noise during Carsten's talk as well. The Q&A, which we encourage people to participate in after the talk, we'll stick around for an hour for that. I'll be moderating that, and what I'm going to do is look in the chat function and I ask that if you have a question, just type a question mark in there, and during the chat function, and I'll call your name as they come in, so that we can have people ask questions. And so at that point, I'll say, please, ask your question to Carsten, and we hope that will facilitate a really interesting discussion later. So with that aside, I want to introduce Carsten Schradin. Carsten, as I said, is the sort of the mastermind behind all this, and it's been really great working with him on this project. Carsten is the director of research at CNRS in Strasbourg, France. He has a diverse research interest. In particular, he's focusing on behavioral and physiological flexibility in African striped mice. This is part of his 20-year study of African striped mice in Africa, where he is working out of the Succulent Karoo Research Station in South Africa, where he is also director. Carsten is also interested in the social evolution and thinking about intraspecific variation in sociality in mammals. And he's now taking some comparative approaches to try and understand this important topic. And Carsten's been really productive over his career. He's had 101 publications in peer-reviewed journals, and many of those, I can say, obviously are excellent pieces of work. So with that, I'll turn it over to Carsten Schradin, and I hope you enjoy the seminar today.
Yeah, so thank you very much for the introduction, and thank you everybody for being here. It's so exciting to have this finally starting, and I can tell you all three co-hosts, we are excited all day already. We're looking towards having some exciting interactions. First of all, I would like to thank the research team of Barbara and Michael Taborski in Bern, who had invited me for a Zoom seminar in May. And this was such a positive experience for me that I said, why don't we do this every week with people from all over the world? And that's what we are starting today. And I'm very happy about this. The other point is that I'm in a kind of midlife crisis. I did 20 years of study on striped mice. And in 20 years, I'm going to retire. So I'm asking myself, where's this all going? And this is also for me interesting. I want to see this seminar series as something where we don't only talk about what we have achieved, but also where we think our field of behavioral ecology is moving towards, because this is not the end of behavioral ecology. The last maybe 5 decades or longer, we started doing exciting field studies. And for me, one question is, where will our field be maybe in 50 years? And this is why we call the series Frontiers in Social Evolution. And I hope that all the people that are going to present here also give some ideas what they think are open questions we still need to answer, what we should do in the future. And my talk today will have 2 parts. The first part is where I will tell you what we've done on striped mice over the last 20 years. And the second part is getting a bit more critical and a bit less clear, more open questions than answers, because I think that's what science needs. We need open questions to move forward. And this is what makes it exciting for us. So today I'm going to focus on mammals, not because I think mammals are more important than any other taxa. We have exciting talks on spiders, on insects, on birds, but it's just because I'm talking about a complex pattern, and mammals is what I know best. And so I want to focus on where my expertise is, and then hopefully benefit from all the other exciting talks coming during the seminar series. So social systems, one of the key questions in behavioral ecology is how can we explain this diversity, biodiversity in social systems you observe in nature? Why do animals live in groups? Why do they show alloparental care and all these things? And as scientists, we always have, when we study something, to define what we're really working on. And we, with this I mean Lauren, Eduardo, myself, and of course many other people, we follow the terminology from Kappeler and van Schaik that was updated by Peter Kappeler, who is today in the audience. I've seen last year in a very interesting and nice paper. So we follow this terminology, and after this, social systems has 4 components. First, the social organization, that's the group composition, the social units, what are they composed of? Group size, also kinship pattern, is the group a family, or is it 2 unrelated males and 2 unrelated females? The social structure, which basically describes how animals are interacting with each other. And importantly, and I will pinpoint this out at the end of my talk, this is not only within, but also between groups. The care system was taking care of the offspring, and the mating system, who mates and who actually reproduces. And all these 4 components can have different states. For example, social organization can have at least 6 states. They can be solitary, pair living, 1 male, multiple females, multiple males, 1 female, multi-male, multi-female groups, or even sex-specific, as we found out in ungulates by studies from Lauren's group, where in some species the males are solitary and the females live in groups. In social structure, there can be many different components. Are there pair bonds? Are they territorial? Dominance hierarchy? Are they egalitarian, or is there strict dominance hierarchy? Cooperation. Care system, most animal species, nobody takes care. There's all these different other kinds: maternal, paternal, bipaternal, communal, or cooperative care. And the mating system, of course, can have different components as well. And when we look at this, these different parameters that are possible, it means that overall there are more than 1,000 different social systems that are possible if we combine these with each other. So there's a huge biodiversity. And to try to understand all this at once is very difficult, if not impossible. And this is why in my research team, we focus on 1 component only, which is social organization, and here group composition, to have less variables within our studies. And for this, I've been studying now for 20 years the striped mouse in South Africa and the Guchab Nature Reserve, which is 600 kilometers north of Cape Town. So typically we fly down to Cape Town and then I'm driving up here. It's a semi-desert habitat. You see it's quite open. Striped mice are diurnal, so active during the day. They occur in this open habitat and are easily habituated to the presence of observers. Such we can observe them directly in the field, as normally is only possible for large animals. And as nearly half of mammalian species are rodents, and most rodents are mice, and as such, on average, a mammal is a mouse, I think it's very important that we also have studies on wild living mice to come to an understanding of mammalian social systems and social evolution. There, I didn't build up, but I established a Succulent Karoo research station. We got this old farmhouse. We are maintaining and developing. The Succulent Karoo research station is a registered non-for-profit organization in South Africa, providing infrastructure to conduct research there. We have electricity, fridges, centrifuge, field laboratories, vehicles, and most importantly and conveniently, it's directly in front of the field site. From the door, which is here, it's a few minutes walk to the first group of striped mice. And since 20 years, and you see here this evolution of myself, so it's rather ontogeny than evolution, and many students have helped us there to conduct exciting studies. I did their own master, PhD, or postdoc projects. I've seen at least one postdoc, Davina Hill, is in the audience here today. And a lot of people benefited, and I benefited a lot from all these people. But of course, unfortunately, due to the corona pandemic, the station was shut down in March. And this is then now the first time since 20 years there's no field work conducted. The nature reserve itself was closed until yesterday. They opened today. And I hope that they will soon tell us that we're allowed to send students there. There's no travel allowed to South Africa, but we have a South African student, a very competent one, Lindelani, who is also in the audience, and we hope that we can send her there soon, that she can do some basic trapping, at least for us. And we very much hope that next year we can start the project again. So what did we find out with striped mice? The first things that I found there is that they were group living. So every circle here shows one group of striped mice. Red are the breeders, bluish are the philopatrics, their offspring, which can remain within the groups even long after reaching adulthood. So typically what we found is one breeding male and 2 to 4 breeding females and the offspring of both sexes remaining in the group long after reaching adulthood, leading to groups of more than 30 individuals of both sexes. And that's what we especially observed under high population density. But when population density was not that high, we also found that some females left the groups and started solitary breeding without any male associating with them permanently. And we also had some solitary males, we call them roamer males, which try to mate both with the solitary females, but also with the females defended by these territorial breeders. And in a year with extremely low population density, we found all adults to start solitary breeding. Both the males and the females started to become solitary. And this is shown here in this graph that shows field data of some of the first 8 years of studies we did in South Africa, the relationship between population density and mice living in group. And what we found is high population density, most mice are group living, low population density, all become solitary living, intermediate population density, we find both tactics. And this indicates that the higher the population density, the smaller the home ranges, we also showed this, and the more mice live in groups, which is in favor of the ecological constraints model, saying that if animals cannot go anywhere, they're forced to form groups and to stay at home. Now, when we add to this, these are data from September in 8 years, the start of the breeding season. If we add data from the non-breeding season, that's before September, that's July or August, and in January, we find that outside of the breeding season, mice nearly always live in groups. And just for you to let you know, the mice up here is the year 2003, are exactly the same mice like down there 1 month later when the breeding season started, showing these mice were living in groups. And then became solitary, also showing they had the option to be group living if they wanted to. But when the breeding season started, they left the groups and started solitary breeding. And this indicates to us that there is something additionally happening in the breeding season that's not happening outside the breeding season, and that's reproductive competition. And in a series of studies, which I don't have time here to show in detail, we could show that there is reproductive competition between males. There's sexual suppression. The dominant breeding male suppresses the remaining males. And especially also in females, there's female infanticide and female-female aggression. If you keep females in captivity together, they kill each other's pups if they don't give birth the same night. And thus, if there are free territories, some of the females decide before they give birth to leave the group and start solitary independent breeding to avoid this reproductive competition, and the males then follow. So these are 8 years of correlative field studies. We repeated this study by my PhD student, Ivana Schöpf, who did experiments where an experimental field site, she experimentally reduced population density, and she found nearly exactly the same pattern like in these correlative field studies. And not shown here on this slide is when she repeated this study in the non-breeding season, mice remained group living even if population density was decreased significantly. Now, this shows that variation over time, over years, leads to social flexibility or intraspecific variation in social organization in striped mice. And one question we had is whether this variation we also find when there's variation not in time, but in space. And that's a study we did in 2018, asking does social flexibility allow an adaptive response to spatial variation? And instead of studying 8 years of the same population, we wanted to study 8 populations at the same time. And for this, we used subpopulations in the Guchab Nature Reserve. This is an area of about 5,500 hectares. Here are different populations. They are separated from each other by sandy flats or mountain passes where no striped mice occur. Because we had some years of severe drought, population densities was overall low and 2 populations became extinct, leaving us with 6 populations which we could study in the end. And this paper I just corrected the proofs yesterday will soon be available in behavioral ecology and sociobiology. And the data from this study on spatial variation is shown here in black, and we found nearly a very similar relationship with these 6 populations during the same time like we found before with 1 population over 8 years, indicating that this flexibility in social behavior leading to intraspecific variation in social organization might not only be an adaptation to change over time in ecological conditions, but also change in space. For example, we had several mice that were dispersing from the main field site to other field sites, and mice that disperse from high population density might find a population with a different population density where they might have to switch their tactic to reach their optimal reproductive success. So intraspecific variation in social organization is a tool to study the approximate and ultimate reasons of sociality. We did many studies on this, so especially differences in hormones. Can be part of the answer to the questions, will populations be able to cope with global change? Can populations change their social behavior and their social systems in response to global change? And most important for me, it's an interesting phenomenon by itself that requires scientific explanation. It's important to understand the social system of a species. To say, okay, striped mice live in communal groups is not correct because they can show different forms of social systems. And this is important because presently the phylogenetic approach to understand the evolution of the variety in social systems is flourishing with many very important papers. And this needs the correct classification of the social system or social organization on the species level. This is not something new. Lott published a book and review papers on this in the 1990s, that there's intraspecific variation in social systems, but we are building up on this. So what is social organization? There are different forms of social organizations animals can show. They can be solitary living, pair living, with or without offspring, group living. Group living can have different forms. It can be one male, multiple females, multiple males, one male, multi-male, multi-female groups, or only males and only female groups. It can also be sex-specific, like in ungulates, we found some species that males are solitary, females are social. And theoretically, though I don't know of any species that shows this, it could be that males are social and females solitary. And we talk about intraspecific variation in social organization, short IVSO, if 2 or more of these categories occur, importantly in both sexes, both in males and in females. So what is no IVSO? There are cases that we don't regard as IVSO because if you would include them, then basically every species would show it, and then this concept would be useless to explain and to describe anything. This is if only one sex has more than one form of social organization. For example, solitary individuals. In all mammals, nearly all mammals, we have solitary dispersers. It's often the males. And if you would count that as a different form of social organization, then every species would show IVSO, and it would be pointless to try to study it. But in many species, for example, in striped mice, we know that there are also dispersers of both sexes. So when we record solitary individuals, we have to make sure if this really their social organization in which they live and reproduce, or is this a dispersal tactic. Further, if there are alternative reproductive tactics in only one sex, we don't regard this as IVSO. This is because in species, for example, where we have one male living with multiple females, the other males must do something. And they often have alternative reproductive tactics because there's equal sex ratio in mammals at birth. So the remaining males must be somewhere. They could also be, for example, in bachelor groups or in female in bachelorette groups. So important that we regard it at IVSO is that both sexes show more than one form of social organization. So IVSO, from my point of view, should be considered to understand the social system of a species, saying striped mice are solitary or they live in one male multi-female groups. From my point of view, is not correct. It can and should be considered in comparative studies on social evolution. And in previous comparative studies that were very important, there were some shortcomings that could be improved in the future because IVSO was often ignored and species were either taken out from the analysis because people didn't know in what category to put them, or they categorized them for one form of social organization only that could have been the most common form or the most interesting form from the point of view of the researcher. Another problem from my point of view is taxonomic inference, that assumptions instead of real data enter databases, meaning that if species that have not been studied in the field have been assumed to have the same form of social organization like closely related species. And I will bring now two examples from other research groups for very important studies where I think considering IVSO could have further improved their conclusions. The first one, and I really love this study, is on eusociality in the snapping shrimp, Synalpheus. I love this because people, when they talk about eusociality, they only think about bee and ants and so on. But eusociality also occurs in other taxa, like in these shrimps. That some species can live in pairs, one male, one female, others in groups with several adult breeding females. They call this communal breeding. That's, by the way, a study by Solomon Chuck from the research group of Dustin Rubenstein. But there can also be eusocial species where there's only one breeding female and other, they call it a queen, and specialized adult helpers that help raising the offspring. And the question they were asking is, did eusociality evolve from a pair living or from a group living that's communally breeding, ancestor, or from both? And they had three competing models for the evolution. That could be that P stands for pair living leads to communal breeding and communal breeding to eusociality, or pair breeding leads either to communal breeding or to eusociality, or pair living leads to eusociality and then communal breeding. And their results were most supportive of model 2, suggesting that communal breeding species do not represent an intermediate form, like it would have been here, of between pair forming and eusocial species. Now here we see all the species they had in the beginning in the database, and from their study they concluded eusocial and communal Synalpheus species each evolved directly and independently from pair-forming species. However, there were some species that are shown here in blue and red that showed IVSO. And from their main analysis in the paper, they had excluded all these species that showed IVSO, which I think is a pity because some of these species show both eusociality and communal breeding, which is kind of in contrast to their conclusion. And by excluding these species, I think they missed something. But in fact, they did not totally exclude them, because if you dig deep into the supplementary information, you find an alternative analysis where they considered these species with IVSO. They call them intermediate species T1, that there are species that show pair and communal breeding, and species that show pair living, communal breeding, and eusociality. And when they included this in the study, their best supported model was model 3. where, not surprisingly, the intermediate ones with pair living, communal breeding, were in between pair living and purely communal breeding species. But here we also have species where communal breeding and eusociality occur at the same time, most likely coming directly from eusociality. So this analysis is in contrast to one part of the conclusion saying that communal breeding only evolves from pair living, because it can also occur in species that evolved from eusocial species. And for me, this story in the supplement is more interesting and is more comprehensive than the story they put in the main manuscript. And I think for me, this is shows that IVSO is very important, and considering it and not simplifying it to get a clearer story might still lead to very interesting results, and results that are closer to reality, because we have these species that show both forms of breeding. Then when we started, and that was Lauren and myself, we wanted to understand IVSO in all mammals. And we were first using as a template the database by Dieter Lucas and Tim Clattenbrock from their Science 2013 paper, where they have more than 2,000 species of mammals and their social systems, and to see if we can use this to understand the evolution of IVSO in mammals. And the first taxa we started with to check this database, how far it suits our own questions, were the Eulyptotyphla. Now, Eulyptotyphla, I'm very proud that in the meantime I can pronounce this word, so I will say it often. Eulyptotyphla were formerly called insectivores. So these are the shrews, the hedgehogs, the moles, and so on. But there has been some new things in taxonomy, like it always happens, and now they are not called insectivores anymore, but Eulyptotyphla. Lucas and Clattenbrock, they had 397 of these species in the database, and now 399, and most of them being solitary, 99%. And my student, Mia Valomi, was digging through all the literature on all field studies that have ever been done on social organization in Eulyptotyphla, and she found that there were data only for, 16 species that have actually been studied in the field in detail for social organization. And out of these 16 species, 7, less than half, were found to be purely solitary. Some, 4, always lived in some social groups, could have been pairs or real group, and some of them are variable with solitary living and group living. And the conclusions from this, that 44% are solitary, is in sharp contrast to this 99% reported in this database. And interestingly, when we compare this to the book Mammals of the World, these are 2 huge heavy books, very big books where people build up all the knowledge about mammals, we find actually that the information reported in there is quite similar to what we have found. So we are confident we didn't miss a lot. Out of this arose controversy between myself and Dieter Lucas and Tim Clattenbrock. It was published in the Royal Society of Open Science. And I want to talk a bit about this here because it shows how important it is to look at how different people define the same terms. So we focus really on social organization, while their studies, and the original study also 2013 in Science, focuses on social monogamy and cooperative breeding. So what does this mean? It means we really are interested in group composition, the composition of social units, but they looked more at the combination of group composition plus mating system, how many breeding females are there per social unit, and social structure. For example, the term pair bond is important for them. And this is something that confused me, because in their method section, they said our classification of species focuses on the social organization, and I automatically thought social organization is the same like we use, but they actually focused on breeding, which for us is breeding system, mating system. So this is something different. Then also how to define solitary. For us, solitary is there's a single individual that only meets other individuals for mating or territorial disputes, otherwise lives alone. But for them, solitary means there's only one breeding female. They focus on pair living, communal and cooperative breeding, and everything they call solitary. So if you have a group of 3 females and only one breeds, for them that would be solitary. For us, it would not be. This is also a different kind of how to define this. So they classified as either solitary, breeding females forage independently. So I understood it's the same like we did. And the main conclusion that we show that the ancestral conditions for all mammalian groups is of solitary individuals they don't mean solitary the same like I do, that it's only one individual, but they refer rather to there's only one breeding female and no breeding male at the same time. Also, they were more stringent. When we read papers and they always observe pairs, one male, one female, then for us, this is pair living. For them, when it is only when pairs observed but not monitored during the entire breeding season, they regarded them as solitary breeding. Only studies where pairs were observed or cross-breeding attempts were recorded as pairs. And so some studies where people report pairs, but they don't report that these pairs stay together for a long period, for breeding season or even outside the breeding season, they recorded them as solitary living and not pair living, in contrast also to us. And last but not least, if we didn't find any study on a species, we didn't enter any data, and they used phylogenetic inference. So they thought, okay, there's no data, but similar close related species has been studied, so we assume the same. So this teaches us that definitions must be clear and terms like solitary individuals must be defined. And when we use a term and somebody else uses the same term, we have to be careful. Do they also mean the same, or do they only use the same term? And what they conclude, which I think is very important and which we should all consider, is before using an existing database, check its definition and whether this database suits your own question. Because many people now look for databases, including this database, and want to use it for their question. But the fact that there's a database that uses terms which you're interested in does not mean this database is suitable for your question, and you might still have to check it. You have to check it always, or you might have to build up your own database, which is what we are doing at the moment. And comparative studies, there has been more criticism over the last few years on the databases used. For example, by Michael Taborski, there has been a criticism on a database on cooperative societies in cichlids. There has been a study published, and they checked the database after it was published, and they disagree with the database in this study on societies in cichlids. Also, Michael Grieser criticized that studies on cooperative breeding in birds mix everything together. Species that always have cooperative groups and species where only 10% of the nest show cooperative breeding. And they argue that these might be very different things. Species where cooperative breeding is obligate and species where it occurs in only every 10th nest, and that one has to differentiate this. And now we spend a hell lot of time getting together this database and social organization in mammals. Of course, we hope later to analyze this with life history data. And so we thought, oh, at least life history data, we can easily take out of some existing databases like Panthera. And unfortunately, Eduardo Fernandez-Duque and Marin Hook and Anthony Di Fiore destroyed my dreams, because they published a paper beginning of this year where they showed that in this database, which many of you might know and many of you might use, Panthera, and they looked at one factor they know very well, home range size in their own study species. What they know and what's written in this database differs by the factor 2, 3, or even 7 times. And that means that even these databases, before you use them, one has to be very careful, because it might not be the most reliable information. So we've been collecting data on social organization in different mammalian taxa, including primates. And primates, of course, are of special interest to many of us, as we are primates ourselves. And it has generally always been assumed that the ancestor of all primates has been a solitary tree shrew-like animal. That has been said by the study by Lucas and Glattenbrock, also by the Schulz et al. Paper published specifically on primates in Nature in 2011. But in these studies, IVSO was not taken into account. They were put into one category, or in the Schulz study, some species they did not know for sure what their social organization is, and then it was partly the species were excluded. And we wanted to, as one of the first taxa, to look at this in more detail. In our new approach, I call it new, we clearly focus on social organization. We avoid a mixture of social organization and mating system or social structure. So we only look on the composition of social units. We only use information from field studies, not from studies in captivity. We also don't use taxonomic inference. If there's been one species of lion tamarine studied, we use this one species and don't assume the 3 other species have the same form of social organization. We take variation into the database. So if there is IVSO, we record it as IVSO. Also, we did the analysis on the population level. So every population is a separate data point because some populations might have a different form of social organization than the other one. And we used a clear definition of solitary living. That means both adult males and females are alone for the entire activity period with or without depending offspring. They might only meet during mating or territorial disputes. So species where the same individuals meet daily, are miscible to each other, share a sleeping place, and defend together territory against other conspecifics were not regarded as solitary in our database. Though in the other databases, because they spent the foraging time alone, they were often still regarded as solitary. And in a more sophisticated analysis to follow, compared to what I show you now, we will take this specific case of group living solitary foragers that we find in many nocturnal prosimians, like mouse and dwarf lemurs, tarsius, and bush babies into account to maybe explain some discrepancies. So this study was done by my master student, Camille Pillay. The statistical support was given by Adrian Yagi from the University of Zurich. We see here the tree. We see every dot here is one population or one form of social organization, solitary living pairs, stable groups, or variable. So that's IVSO. And so if there's for one species more than one point, it means that more than one form of social organization was observed. For example, here we have a species that was both pair living and variable in two different populations. And the most likely ancestral form for the primate ancestor was found to be variable and not solitary living. Actually, solitary living had the least support. Now, it could be that variable here means that solitary living is part of this variation. Maybe this is a lot of species that can be pair and solitary living or solitary and group living. So we split this into more subcategories, and this is what we see here. This is all the variable forms: solitary pair, solitary group, solitary pair group. All these three variable forms have nearly no statistical support at all together with solitary living. And so it is most likely that the ancestor had a variable form that was kind of sociable and not solitary living. And we're at the moment improving the database and the statistical analysis, and hope to have a nice study coming out of this next year, then together with my new PhD student, Charlotte Arnay. So generally, I grew up with the belief that the mammalian ancestor was solitary. I always thought our ancestor was this small nocturnal mouse-like animal running around solitary between the feet of the dinosaurs, trying to survive, waiting for them to go extinct and then become more sophisticated and social. And with these studies on the shrews already and the primates, I started to think, why do we believe that actually? Is this really true? And for this, we are now building up a database on the social organization of all mammals. And we already made quite some progress here. In green, you show the taxa that we are finished. In blue, the taxa where we're currently working on. We hope to finish this by the end of this year. And this table shows you an overview of some of the major taxa. This is done in collaboration with Lauren, whose students also contribute to the database, and Professor Adrian Yagi in Zurich, who is will help us with the statistics. Now, what I want to point out here is the number of species studied compared to the number of species reported by the IUCN is often very low. So for most mammals, we really don't know yet how do they live in nature. Especially interesting for me are the opossums. In opossums, out of 98 species, only 8 species have been studied, and most of them are solitary, and people actually believe all opossums are solitary. Now, opossums are small animals. They are nocturnal, arboreal. They eat insects, are omnivorous. So they're nearly like the prosimians of South America. And prosimians were believed to be all solitary some decades ago until people started to study them. And if I could recommend something, is study some of these opossums. Maybe something exciting and you will be found in the way that was discovered in the prosimians when people started to pay close attention to them. Still, IVSO is very common in most of the taxa. Actually, opossums are one of the few taxa where it's less than 50% of the species, also in the odd-toed ungulates. And we start to think about it, maybe rather we have to think how stable social organization evolved than IVSO. And solitary living is relatively rare. Also, again, opossums are an exception, where it seems to be from the few species studied more common than group living, but it is not very, very common. Now, going back further in time, now when we see this mammals that have been studied, solitary living is not that common. I was looking into what is known from paleontology, and there's some interesting studies on the known mammalian form cynodonts living about 250 million years ago that gave rise to mammals. So these are the ancestors of mammals. And what was found is that they spent part of the time living in burrows, and associations of both juveniles and adults, as well as several adults, were found in fossilized burrows, indicative of a social lifestyle in this, the mammalian ancestor. And the absence of sexual size dimorphism in one genus, Trinaxodon, these are these guys down here, and a small number of adults found together, was interpreted as indication of pair living by this paleontologist. Now here from this paper published 2017, there is this table I put out, and I only want to emphasize this here, that on many of these fossil founds, what they interpret these paleontologists is that there was parental care aggregation. That means that there was groups of animals living together and not solitary living. Here you see some of these fossils, and these are typical fossils. These are not the exceptions. Here we have this Galeosaurus fossil. You see here the real fossil, and here a drawing. There's a yellow individual, a blue individual, and a green individual found together. And this is what they typically find, several individuals together. Now here you see these two Trinaxodonts, a pair of Trinaxodonts together, and it's difficult to assume that this was a purely solitary animal. Also, burrow systems have been found, and in these burrow systems, often entire groups of individuals of these ancestors of mammals have been found together. Then we have to make a big jump from 250 million years ago to the next bed of fossils. That's 60 million years ago. So dinosaurs were already gone. Now mammals could do what they want. And this is a study on a marsupial, a possum-like marsupial from Bolivia. And here they also found in these fossils that often many individuals, adults, subadults, and juveniles were found together, also indicating that this very early marsupial might have had a gregarious or social form of social organization, might not have been solitary living. And this is only 5 million years after the dinosaurs have disappeared. So we always think something like maternal care, paternal care, group living, and pair living is something very sophisticated, far advanced, evolved, something that occurred late in evolutionary history, somewhere at the end, because we are maternal, paternal, pair living. So that's the thing that came last. But is this really true? Did this occur late in evolutionary history? Now, of course, at least for maternal care, this occurs in all mammals. We can be sure it occurred in the ancestor of all mammals as well, at the first mammalian forms, but maybe even in the cynodonts. And paternal care, is it something new in some rare mammalian species, or did it also occur early on time? It's difficult to say. And for group living, people assume the ancestor here was solitary living, but there's indication that already the cynodonts were not solitary living, and thus group living might have occurred much earlier in evolutionary history than is so far believed. And for pair living, it's the same. I mean, I showed you that some paleontologists believe that already some cynodonts were living in pairs. Then comes the question, when did solitary living evolve? So far, people think the ancestor of mammals, either here or here, was solitary living, but maybe solitary living is actually something advanced. Maybe solitary living instead of group living evolved later in mammalian history in some specific taxa for which it is of advantage. And just to show you that it's not only social evolution, but other parts of mammalian evolution that get a change, I bring here the example of torpor and hibernation, which also was believed to have evolved very specialized very recently in time. But Barry Lovegrove, in his book Fires of Life, and he has a review in Quarterly Review in Biology, argues that torpor was actually at the very beginning of mammalian social evolution, allowing individuals to survive harsh environments. And just a few days ago, there was a paper published of this saber-toothed small mammalian ancestor from Antarctica, where by looking at the teeth, they found that this animal already showed extended periods of torpor 250 million years ago. So torpor and hibernation might be an ancestral state in mammals and not something very derived. So this could maybe lead to some transformative science, changing the way we see our own ancestry, at least the way I see it, because so far I believe we have this solitary ancestor running between the feet of the dinosaurs, but maybe the ancestor of all mammals was actually much more sociable than I believed so far. It could be, of course, that there is a bias in studies on mammals that's in our database, that it's mainly mammals that have sophisticated forms of social organization that are studied, pair living ones, eusocial ones, group living ones, and maybe our database, because of this, is wrong. So I think there are three main reasons why somebody studies mammals. First, charismatic or important species that just look great, like polar bears, leopards, elephants. And already, of course, I want to indicate to you that this does not exclude solitary living species. And if there are species that are believed to be solitary living, but they are important for us because they're primates, then all of a sudden, when we do detailed studies on them, we find that Tarsius and Caryogaleus are not solitary as believed, but they live in groups or in pairs. Many people study species living next to the university simply because it costs a lot of time and money to go to the field to exotic places. So they study what is next to their place. This includes this shrew Suncus variella in South Africa, which was studied by researchers close to the University of Bloemfontein, and they found it to be pair living. Another species, Rhabdomys dialectus, a sister species of my main study species, Rhabdomys pumilio, is in fact solitary living, has also been studied because they occur in grasslands next to universities. Then we all know prairie voles, model for pair living or social monogamy. They're also studied next to universities, and next to some universities, they're not pair living, but rather solitary living. And the same here in Europe for the Microtus avalis, the common vole, that also has a rather variable form of social organization, including solitary living. So I think this doesn't exclude the study of solitary living species. But then, of course, there are some people here in the audience that only study the most interesting species. That's especially myself, studying paternal care and group living in striped mice, Lauren studying the colonial degus, and Eduardo focusing on the pair living old monkeys. And of course, these are the guys who put the focus maybe wrongly on species that are only very exciting. And here I would like to use this talk to advocate that we must also consider solitary species as interesting in the future and consider them, and maybe also people on the field side spend some time and resources and students to study these not so exciting species, because you might find something that is very exciting. So why might mammals be solitary? If they are, it could be in a derived state. In top predators, it might be that they don't benefit from group living as protection against predators. They might need large territories to find enough prey. And in small mammals, it might occur especially in insectivorous species that can't share food. And it can be that in these small mammals, living in groups might more easily attract predators, but you're so small you can't protect yourself against a predator. And this is the reason why in some species like striped mice, like nocturnal prosimians, they have the tactic of group living solitary foragers. They form groups, so they benefit from group living, for example, sharing a nest, but they go alone for foraging to avoid specific costs. But for me, the question is a solitary lifestyle really simpler? Or are there specific difficulties to overcome, and other costs and benefits of group living? And for me, this is one of the interesting questions of the years to come. There has been a very nice study published on the cougar or puma, I think 2017 or something like this it was, yes, here. And here they showed that there is actually social structure within this clearly solitary species centered around big and dominant males that are associated to females and also juveniles or smaller males. And when they meet, at prey, and this has been studied by GPS collars and camera traps, when they meet at prey within, for example, this violet cluster of this male or the red cluster of this male, then they interact relatively unmissable with each other. But if they meet a puma from another cluster, they are highly aggressive. And this shows that even in this species that clearly has a solitary form of social organization, the social structure is quite interesting. And this is also shows why it's important to differentiate between these different aspects of social systems. It might be solitary living, but still have a very interesting social structure, meaning solitary individuals are not per se unsocial all the time. And this is something I want to focus in the coming years, because on my field site, apart from the striped mice, we have the bush karoo rat, which is also nicely for us a diurnal rodent, which we can observe easily in the field. And this is solitary living from all the studies we've done so far. It's bigger than the striped mice. We can put GPS collars on, and we want to understand in the future, why are they solitary? We want to understand the mechanisms. Is there aggression by the mother forcing juveniles to leave the maternal nest, the maternal territory, and become solitary? What are dispersal strategies? How is this influenced by hormones? From the evolution, what are benefits of being solitary? What are costs? And how do they solve social problems? For example, we expect that even though they are solitary, there is female kin clusters that neighboring females might be closely related, and they might behave very differently to a neighbor that is a close kin than to a neighbor that is a stranger or non-kin. The environment, of course, will be important: population density, competition for food, and competition for nesting sites. And this already indicates that when we go to this step to understand solitary living, the social organization of solitary living, then to understand the social structure will become very important. And for this, we're actually looking, Neville Pillay, who is the co-investigator on this, and myself are looking for a postdoc to start next year. The postdoc would be based at the University of the Witwatersrand in South Africa, but should be dedicated to lots of field work in the field. And we hope to have the funding. The funding has been approved, but because of the corona pandemic, there can be some funding cuts. There's some uncertainty. We will know this within 1-2 months. But if anybody here in the audience is very dedicated, hardworking, and interested, he or she could please contact me. So to conclude, from my point of view, we might have to reconsider social evolution in mammals when we base our analysis on data derived from field studies only. So we don't do taxonomic inference. We only consider species that have been studied in the field, and when we consider IVSO instead of putting them into one category. Mammalian social systems are very flexible. Our reviews of, in the meantime, thousands of species shows that IVSO is very common, and it might be more interesting actually to explain in the future why stability has evolved. And most mammals are social, and few of them are solitary living. And the solitary living ones might still have a very interesting and complex social structure. So solitary living in mammals might be a derived state, not the ancestor state, representing an adaptation to special environments. So future studies should be done on the costs and benefits of solitary living. So this is always the conclusion. So somebody should give me money to do it. But my conclusion for you, or what I would like to ask you, is please invest into studying the social system also of unspectacular species at your field site or next to your university, not only the ones that look to be very interesting. And if we want to understand social evolution, if you think this is important, then we have to collect this data. We're not at the end of behavioral ecology. We have much more data now than 50 years ago. And in another 50 years, I hope we don't only have many more R codes, but also more field data. And last but not least, if you want to have scientific fame, then you have to do this. I'm sure in 50 years, nobody will talk about my papers on alternative reproductive tactics, on paternal care. But if somebody has an online database or whatsoever on social organization, my basic studies on social organization and striped mice will still be studied. So this is more something for long-term scientific eternity than these theory-based studies. And as such, I think these studies are really worthwhile, and we should all invest into this. Thank you very much for your attention.
Well, thank you, Carsten. That was really a fascinating talk. I really enjoyed it, of course. So at this point, let me just find my chat function here. I am notorious for losing it. And then we'll start to have a question and answer. If you have questions, please just type in a question mark and I'll call on you and Carsten will engage. Carsten, am I set up to receive questions here? I'm not getting any yet.
I changed it now.
Okay.
So now it should be possible for everyone to chat. Oh, now it's publicly. Before it was host only.
We can do it publicly if it's easier.
Yes, and then everybody can see it.
Okay.
No questions? Now they come in.
All right, we'll start with Eduardo.
Thank you, Carsten. Thanks for a wonderful talk. Thanks everyone for joining us. We look forward to seeing you all again this next many weeks. And Carsten, I liked it how you finished the presentation speaking to our younger colleagues in the audience. I anticipate neither you or I will be doing field work for another 50 years, not even if I live that long. But the unspectacular species, you're absolutely right. It is a challenge. So many, the choice of species, contrary, we would like to think that is based only on what may give us the best system for studying, but so many times is constrained by other factors. I mean, you mentioned proximity to the university, funding is a big one. And more I anticipate that funding, or a big chunk of the funding, is going to be associated with species that are in danger, however you want to define that. So what are things that you said that let's have some time for talking over the next few months about the future? What could we be doing to continue working on changing that landscape? I'm thinking of Tim's Clattenbrock paper about supporting long-term research. It's been already 10 or 11 years that he published it. I myself, I wanted to study howler monkeys in Argentina, which is maybe among primates a less spectacular taxa, and I was discouraged from doing it because it wasn't in danger. It was, I mean, so what are things that you, since we're just trying to think together, what are things that we could actually do in the next many years locally at our institutions to get this message across that we need funding for basic natural history data from as wide a range of taxa as possible, regardless of whether they're going to make the cover of National Geographic or not. And maybe I'm being unfair to National Geographic, but I use it as an example that we cannot relate to.
So I think the senior scientists have to make an example. Of course, it's getting money is like pressing blood out of a stone, but the most expensive thing is to find time and money to have a field site. And if you have a field site with one of these very exciting species, then get a master student to work on a less exciting species on the same field site, and he or she might find something that is very interesting. So we also, as the more senior scientists, have to get students working on our field sites, which is the most expensive thing to set up, on the field station to work on less dramatic species. And I'm sure something interesting will come out, and this might then open future avenues to find funding. So I think that's the first step to do. And once we have done this, and I mean, then we might approach associations like the American Society of Mammalogy to have especially grants for this, have other funding agencies. And I think it's a very good time to do so now because, like I said, I think social structure is very important for many of these species, and social network analysis is another part in our field that is flourishing at the moment. And we have now some statistical tools which will help us to find fascinating patterns which we could not have done 20 or 30 years ago.
Okay, thank you. I have a the question coming in from, I think it's Radispiel. I apologize, I don't recognize the.
Yeah, hi. Hi, it's me, Ute Radispiel from Hanover, Germany. Thank you very much, Carsten, for the very nice presentation. Quite some very intriguing examples and analysis that you were talking about. I have a question regarding your own work, and you were starting with these interesting dynamics that you have on your striped mice, depending on your population, varying population densities in your study area. And I was wondering if you could elaborate a little bit more about the costs and benefits of these different strategies under these different density regimes. So did you, I assume you did, but you were not mentioning it, did you measure some fitness estimates, how well the females are doing under each of these regimes? What is driving this dynamics?
Yes, for the females, we don't have all the data yet. We have been studied best in the males, and in the males we have three tactics. We have the breeding males that defend a group of females. We have the roaming solitary males and philopatric males that stay in groups. Philopatric males have very low reproductive success, but they have some, they try. Now, the interesting thing is when you compare the breeding males, the group living ones, and the solitary males, what we find is at the high population density, the breeding males have 10 times higher reproductive success than the roaming males. Under intermediate population density, they have equal reproductive success. Under very low population density, we have the problem there are no breeding males, but there we find that some of these roaming males have extremely high reproductive success. So that shows that being a roaming male, depending on the environmental conditions, can mean you do way above average, you do average, or you do way below average. And so this really shows that these tactics can also be the consequence of different things, either doing the best of a bad job under high population density, or doing maybe a very good job under very low population density. For the females, unfortunately, we don't have a good comparison yet of reproductive success of solitary versus group living females, but we had been working on this.
That's a kind of a fascinating bit on the side, because of course all the models predict that the females are driving the spatial structure and also then determine what the males can do best. I mean, if you're widely distributed, the females are widely distributed in space, then it may not be a very profitable strategy anymore to monopolize one single female or whatever, and then it's better roaming around and trying to get access to as many as possible. And if they are more clumped, then they may be more easily monopolized. But then the question is, what is the female? What is going on the female side? Do they have equal success on each condition? Or you mentioned something like there can be infanticide on the side of the female. So I was just wondering whether there is some dynamics coming from that end as well.
Yeah, exactly. Unfortunately, our collaboration doing the maternity analysis on the striped mice has not been as good as hoped, but we have the evidence there is female infanticide and in captivity that can have heavy costs. And we also have the evidence from studies by Davina Hale, who is in the audience, that it's mainly the big females that leave the groups to breed solitarily. So it's the one that are more competitive ones, and they might then have the advantage that they can start breeding earlier. They don't have to wait for other females to give birth, so that these females are maternal, not infanticidal anymore, but they leave and start earlier reproduction. But the real study showing the fitness consequences directly, unfortunately, we have not done yet.
More work to do in the future.
Yes.
Okay, just give everyone a heads up. We had a little technical glitch at the beginning, so I'll try and get questions as they're coming in. So if you sent one to Carsten, please just send it again. Apologize for that. This is why we go first in the series. Next is Andres Link. Andres?
Thanks, Carsten, for that nice talk, very enlightening. I wanted to ask you a practical question. For some of us who have field sites, and you were mentioning that there's few data on a lot of taxa, what would you think is the basic study or study design that we need to do? And I just ask because there's a lot of undergrad and undergrad students wanting to do work. I have, for example, putting some colors on armadillos, but they want to look at home range and still very basic things, but maybe we can go further. So if you can tell us exactly what to collect.
Yeah, so for the armadillo, I mean, we had this discussion a lot with home range data, and of course you need to to trap individuals regularly. You have to mark them individually. If you can put GPS or radio collars on, it's perfect. The important thing is report data where these animals are sleeping and whether they're sleeping alone or not. My students, they find a lot of papers that describe home ranges and there's overlap or not. But when there's a lot of overlap and you don't know, do they share nesting sites all the time? Or do they always sleep alone? You cannot really say this is now a group or these are solitary individuals. So the important thing here to add would be data on sleeping sites and whether sleeping sites are shared or not. Unfortunately, when they're not shared, often people think it's not worth reporting, but that means we who are building up a database, we cannot decide whether they're solitary or not. So in this case, I think sleeping sites would be very important.
Okay, thanks.
Okay, so next up, Zulema.
Yeah, hi. That was a wonderful talk, Carsten. I have I really liked your emphasis at the end on the need to study so-called solitary species, because I think that many of the species that we tend to think of as solitary can actually have extremely complex communication networks or kin recognition networks. And I'm thinking specifically as one case of Jan Randall's work on kangaroo rats, where even though each burrow has one individual living in it, it seems like they're constantly communicating with one another, they recognize neighbors, and so on. And I'm actually even wondering if we need a new term. Like, you know, we've had the term asocial, which I think is a very bad term. Now we have solitary, but most people, when they hear solitary, think of individuals sort of living by themselves and interacting perhaps only for mating. And so I wonder if you have any thoughts on that. Do we need, I mean, I think we have different concepts of in this, you know, spectrum of social behavior. And I'm wondering whether you have any thoughts on whether perhaps there's different terminology that we could use for solitary mammals or other species that, despite living alone, in a sense, have this very, very complex social interactions on an ongoing basis.
Yes, thank you. I think you're totally right. To live solitarily might not be easy, and your social interactions might be even much more complex than group living species. And for the terminology, I just think, for me, it's really following the terminology by Kaplan and Van Schaik and saying they're solitary from the social organization, but their social structure is complex, and thus solitary species are not per se unsocial, and probably there's no unsocial mammals. So for me, it's just important to always keep very strictly to this terminology and be clear. And actually, I would not use the term unsocial at all, but then rather say solitary species can be social with regards to the social structure as well, and then to emphasize this. I have a bit more of the other problem. It's a couple of papers I reviewed over the last 1-2 years where people observe in solitary species complex social structure and then say they are not solitary. And I think they're still solitary from the social organization, but just by always differentiating between social organization and social structure, we should get clear, and we have to be clear there. And that's also when somebody gives a talk, she should define what is really meant by these different terms. And if it's not, then one should ask. I think that's important.
Okay, next is Peter Kapler.
Hi, Carsten. I really appreciated your pleading for studying solitary animals, as you can imagine. But I actually have a question about the other main pillar of your message of your talk, is the study or the consideration of what you called IVSO, which I think is important and clearly something that needs to be included in future comparative studies. But I think there are more question marks about the appropriate methods for doing so than you were able to discuss in your presentation. And given that you had a closer look at the snapping shrimp study, and you also had a master student doing a study on primates where this information was included, I was wondering whether you had any advice, on how to really classify species, whether you use variation in space and time of social units on a local scale, on a population scale, on a species scale. You know, how do you use this information to finally classify the species? And then more practically, there's also a question about the statistical procedures that are available in comparative studies of dealing with, you know, multinomial variables or maybe continuous variables. And so any additional information on that would also be useful.
So thanks for the question. It's difficult to answer it in detail. So for the statistics, I have to say, I'm very happy that Adrian Yagi, who's using sophisticated Bayesian statistics, is in our team in the meantime, which we didn't have when we had the paper on prosimians. And I have to rely there on his expertise, and I'm not the expert to answer on this. Then the problem with the classification, this is a very broad problem. So I maybe want to point out one specific thing here we are looking at. Maybe this answers it partly. So we look at the population level, and in the primates, the first taxa we were working on now, what we want to do is, when is IVSO important? Let's put it, we have in a population 10 groups. It's a difference if 10 out of these 10 groups, 6 are multi-male, multi-female, and 4 are pairs. Then when 1 is a pair and 9 are multi-male, multi-female. And in the primate database, that's what we're working on now. It's also why it's not published. We want to run a multitude of models where we have IVSO criteria. Does it have to be 1%, 10%, 20% of deviation from the main social organization that we find important aspects of IVSO or not? So maybe, I don't know if this answers part of your question, but this is one of the main problems we have. If we have 20 units and 1 is different, it's something different than if out of the 20 units, 9 are different from the main social organization. And this we will address in the primate study to look at this problem of categorization.
Yeah, I think that's exactly where we need general agreement and, you know, maybe more explorative studies to see what the consequences are of different choices in terms of classification. And yeah, but this is clearly an area where you can spend some of the remaining 20 years of your career exploring things.
Yes.
Thank you.
Okay, so thank you for that. And our next question comes from Zimei Li.
Hi, hello. Thanks for the great talk. So I have a question on the IVSO, because it seems that IVSO is a common feature for many mammal species. So I'm wondering if it's possible that, like, for some species that does not present IVSO, and only certain form of social organization can be expressed because of environmental constraints, but they are capable of presenting multiple social organizations. If so, how are you going to solve this problem in comparative studies?
Okay, I'm not sure if I understood fully, but I mean, there are 2 important points here, I think. First, originally we wanted to study when did IVSO evolve, but now it seems we might rather in our to come comparative studies have to study why did stability evolve. That's the first point. So stability might be more rare than IVSO. The second point is, of course, we can only use the information that's available. We don't know whether the social organization would be different, if the environment would be different. Behavioral ecology tells us it probably should, but we cannot put any assumptions into our database. And the only thing I can say here again, we need still, after 50 years of field studies, we still need people investing time and energy into field studies to get this information, because what we don't know, we cannot know.
Okay. The next question comes from Ivana Salas Leon.
Hi. My questions goes more about the ancestor for mammals. That you said that mammals have an ancestor that used to have social behaviors, and we might think that evolution leads to more complex organizations in societies. So changing this point of view, should we expect that now we see more solitary living species, or are they already present but we are not seeing it because we are not paying them attention?
So first of all, I'm not sure if I got the question right. One thing I would like to point out is that the reason why group living evolves are benefits of group living. And benefits of group living is nothing that evolved. If benefits of group living exist, they will also have existed 250 million years ago. So there's no reason to expect group living is more complex and has to evolve at a later stage if the benefits of group living are driving it. So these benefits, whenever they occurred, and there's no reason they shouldn't have occurred early, it could have led to it. Then the second question, whether there's more solitary living species. I did not fully understand. Do you mean because we did not study the solitary living species, or did you mean it that in phylogenetic time now there are more solitary living species than, let's say, 200 million years ago? Can you be more precise?
Okay, I'm gonna try. I meant that maybe if we are seeing the appearance of solitary species after our ancestors, maybe should we expect to see more solitary living species in the future?
So if solitary living is a special adaptation to specific environmental constraints, ecological constraints, then we should not per se see more solitary species if these ecological constraints don't become more common. So at the moment, I think we have a very little understanding what are the benefits of solitary living. And without having this understanding, I think we cannot answer your question. And this is what from my point of view, we should do in the future to find exactly an answer to what the question you just asked.
Okay, thank you.
Okay, next is Leah Prox.
Hi, I wanted to ask if the categorization of social organization into 6 categories might not be problematic, because limiting ourselves to 6 categories, I think we might lose information. And I would explain this with an example. For example, when I was looking into social organization data, which was available for rodents, for example, I found there was very little information on the relatedness, for example, of the individuals. And I mean, this is one part of composition as well, right? If you've got a group of 2 mice with some adult offspring who are staying for a while, this is something for me entirely different than if it's unrelated individuals. And then often also the information on seasonality is lacking. So would we maybe categorize mice which live like that into socially flexible when we observe them in pairs during the non-breeding season, and then in groups during the breeding season, but because we don't know how they're related, we assume they switch to group living. And then also, I think another example would be large groups where we can distinguish between really stable groups which stay together all the time, and then groups which have a high degree of fission fusion with exchanging individuals, which I think is fundamentally different from a large stable group, for example. Have you considered that?
Yes, all these 3 points are very important points. The easiest to address is seasonality. In our database, we include whether the social organization has been observed in the breeding or non-breeding season or in both. And we will then in future analyze this separately and see what is influences. Like in striped mice, it's obviously very important. Another example are passerines. Passerines have a totally different form of social organization in the breeding season than when they're migrating. So it is very important to consider this. The second point is kinship. I mean, if you have multiple males, multiple females, and it's one breeding pair and their adult offspring, or if it's several breeding individuals, of course, it's something totally different. And here, again, it would be great to have all this information, and it will also be important, taxa specific, to look at only species where this information is available. It was, for example, a dispute between Schulz et al. And Lucas and Clattenbrock with the categorization of some of the callitrichids, where Schulz et al. Categorized them as group living, and Lucas and Clattenbrock as pair living because they were paired with their adult offspring. So this is very important, of course. And again, the problem is we have very little information here. We have some information on a few species, but on a broader scale, this information is missing. The third point was group size, which is also one component of social organization. And again, it's very important. And I assume that Aaron Sandel will talk about this in more detail in a few weeks. And again, I can only say we try to use the information that we have and to be very strict on this. And information we don't have, we cannot consider. And in these cases, we then don't use the species. But the main problem is for what we have at the moment in our database is we cannot always differentiate whether it's a family group or whether it's a multi-male, multi-female group. So that is something else. There was this question with the armadillos. The armadillos probably, you won't find this, but somebody else studies a non-charismatic species and finds there are groups. This would then be the next step to differentiate, for example.
Okay, the next question is from Steve Dobson.
Sorry, I had to unmute. Thanks for a really great talk, Carsten. That was really interesting. I have a couple of quick questions. One is, you talked about the evolution of classes, like when you were looking on the tree, different classes of social structure, social behavior. And I was wondering how comfortable you are using those classes as traits, because in an evolutionary analysis, what changes are traits, and the classes seem to me to subsume large numbers of traits. That's the first question. The second one is, I was sort of impressed with how much variation you showed us on the phylogenetic trees over the different species. But then I started wondering about sort of the ancestor and what trait the ancestor had. And if there's evidence for a lot of variation at the tips, that suggests really rapid evolution from one state to another. So how comfortable are you sort of back figuring what the ancestor was most likely when your evidence says that there's a lot of evolutionary change in your characteristics?
Thanks, Steve. Can you clarify, what do you mean with classes?
Well, you had the probability of these different classes, like solitary, different forms of group living, for example.
So I think the problem you are pinpointing add is that what evolves is not group living, but what evolves are individual tactics to associate with a group or not. Is this what you are addressing?
Exactly.
Yeah. Yeah, I mean, of course, it depends on how do we interpret our results. And of course, what you're right, the trait we are studying is not the individual trait, but it is the product of individual traits of the individuals within a population. Mm-hmm. This is right. Yeah. So, but I mean, we can still study and understand the outcome of these individual traits in social behavior, as we do have the data for this. This is like every kind of behavior you observe is the outcome of the physiological system below it, the central nervous system, the endocrine system. What has evolved is not paternal care per se, but what has evolved is that some brain areas get more responsive to prolactin, and prolactin secretion has been a level of selection and has been preferred by selection. So even when we look at, let's say, only paternal care, what actually happens in the individual is not there's a switch paternal care or not, but it is something below it. And I see it the same way, like when you look at some individual behavior, it's a product of physiological changes. And if we look at on the level of the social unit, then it's the product of individual behaviors. Then the second question about when you say we observe a lot of variation, it might indicate rapid evolution. I'm not sure. One problem we have discussed here, maybe that's related to it. There's a difference if for the ancestor we find that the most likely ancestral form was IVSO, or when it is not resolved, because all the species that are in our database show something else, then we might find we cannot resolve the situation at the base of the tree. And here the question then that is then raised is whether we really have to study it on the population or on the species level. Like if you have two populations of one species, one is pair living, one shows IVSO, then you cannot make a statement about what was the ancestor of these two populations, and then one might have to go back to the species level. But I'm not sure if this was really your comment.
Well, I'm just thinking that if you have evidence of a lot of flexibility in those things, if you look back 60 million years, if it's really reasonable to think that that you can get an answer. Whereas if you see there's a lot of phylogenetic inertia in something, then it's a little more reasonable to think that you can do the back figuring about the ancestor. It's the same problem of looking at very narrow taxa versus really broad taxa.
Yeah, I don't have an answer to this question. I have to think about it.
Okay. Well, thank you very much.
Next, thanks for that question. Next is Eduardo. Do you want to, Eduardo?
Yes. Well, no, I'm not sure if this is a question as much as we've already been talking and doing exactly what we hope we would do, of hearing from lots of people and thinking together about the things we've done and things we hope to do in the future. So, Carsten, I wanted to ask you, I go back to something we discussed during the Q&A now 10-15 minutes ago. We had quite a conversation about definitions. And I don't need to explain that all three of us at least agree very dearly on the importance of being explicit about definitions. But then there was a question by Peter that got me thinking that in sharing some of our interests and concerns, maybe with the students who are getting started, the people who are launching their new field sites, definitions are going to keep getting reviewed again and again, and somebody's going to come after Kapper van Schaik, and somebody's going to come after Hook et al. And 10, 20 years from now, somebody's going to do some new set of revisions, adding new comments, etc. What I think is a challenge that I've seen in our own project is that with the new technologies, the process of synthesis, the process of summarizing information that goes from what we observe to what persists over time, it's switching where it occurs. It used to be that you would write lots of comments or you would record, right? You would be taking notes with your voice. And so the process of summarizing would happen later. And the definitions would be made later, but you would still record as much data as possible when things are happening. Something that I've seen in our own project is that because of technology, we're making those calls, we're making that summary more and more at the early stages, sacrificing, forgetting, and never recording for future generations the richness of the natural history data. So as much as it is important we define, I think that we can always redo analysis with any definition we want if we have the data, if we have the proper metadata supporting why we say 2 adults and not an adult and a subadult. So I wanted you to comment on that. I know that you're very, very meticulous and thorough and detailed in collecting the data, but as we share our thoughts with the younger generations, I feel like that's where we can still do much more. Be thorough with data collection. Time for defining, time for classifying will come later at data analysis, but not go fast, not skip the richness of the details you ought to collect with your data.
Yeah, so I mean, I totally agree with your point that papers must report real data, which people then can put into their categories, into their definitions. So it's not enough to report interpretations, but that there's a real data table. For example, in our IVSO project, I always tell the students, don't look at the discussion, look at the results section, look at the table, because that's what we want. We don't want what they think. We want to see what they recorded. Then the other point, I'm more critical, the idea to record everything and somebody will then later look at it. I know one example where unfortunately doesn't seem have to work. A famous scientist who was following this approach, maybe outside of Germany, he's not well known. It's Irenaeus Eibel-Eibesfeld, who was working on human ethology, and he recorded tons and tons of human behavior on film, always with this idea to keep it and somebody will later look at it in detail and might interpret it differently. But unfortunately, so far, nobody ever did. And so I'm not sure if keeping all these details and hoping somebody in the future will look at it, that this will work in reality. But I really think it's important in the results section to report the real data, not the interpretations. And then people, if they have a different form of categorization, they can use this to categorize it the way that suits their system.
Zulema, you had a comment.
Yeah, my comment relates right back to what Eduardo asked and the answer. It seems to me that in a sense, what we're also talking about here is how to do science. And there's such a huge emphasis right now, at least at my university and lots of people I interact with, about doing hypothesis-driven research where you have to come up with your hypothesis ahead of time, and then you go out and you collect the data specific as it presumably relates back to your hypothesis, versus an approach. And I don't think it has to be as extreme as Eibel-Eibesfeld, but I was thinking of like the Kyoto School of Primatology, where the approach is very much that you go out and you collect data on these monkeys, everything you see, and then you come back and you go through your data, and then you begin to see patterns, and that leads you to your hypotheses. So it's two different ways, I think, of approaching science. And my own take, I guess, is that I think there currently there's an overemphasis on starting out hypothesis before you go out and collect any data. And I think that part of what it has done is that it has sort of downplayed or devalued natural history and a different way of looking at science consenting data, and then allowing the data that you have to drive your understanding and your interpretation of what is going on. So I don't know if anybody has comments about that.
So I would just say I agree with what you said. And the main problem is if you have to have a hypothesis, you might miss the non-spectacular species, and then you might miss coming up with spectacular hypothesis. So I think we need both. And in the long term, the more descriptive studies will last longer than the hypothesis-driven ones. It's also my belief, because I don't think anybody will cite my paper on alternative reproductive tactics in 20 years. But about the social organization in striped mice, they will.
So next is Marlene.
Hello. Yeah, thanks for the talk and the discussion already. I learned a lot. My question is related to IVSO again. And when you talk about IVSO, do you refer to variable systems between populations, or would you also include flexibility within individuals? So that, for example, long-living individuals are able to adapt to different or to changing environments in order that they change their mating system or their group structure.
So in our database on mammals, we differentiate whether the variation is observed within or between populations. And obviously in striped mice, we see it within populations. And in other paper, we came up with 4 mechanisms that might lead to IVSO. And the most common one seems to be social flexibility, where actually individuals can switch their behavioral tactics. And with this, the social organization of the population can change, which seems to be one of the most common one. We don't have good empirical evidence that 2 populations of the same species differ genetically from each other, leading to different forms of social organization. But yeah, it's important to look whether the difference is within or between populations, especially later, to find out what are the ecological factors associated with this.
So there are no questions in the queue right now, but I want to encourage people, if you have follow-up questions or comments, please, let's keep the conversation going. I'm enjoying it immensely. And also I want to point out, some of the students out there, please feel free to ask a question or make a comment. This is a learning experience and we want you to grow intellectually. So I just had one come in. Nancy Solomon, please go ahead.
You're still muted, Nancy.
Okay, can you hear me now? Sorry
Yes.
about, but I just started to say thanks for a really interesting, thought-provoking seminar. It's really nice to be able to interact with everybody intellectually after this long COVID drought. My question for you, Carsten, is just wondering, when you're doing, taking these phylogenetic approaches and finding that a number of taxa, number of species have, as you say, IVSO, as their social organization, the way you described IVSO sounds like it might not be a unitary thing. So if I understood correctly, if in one species, the males and females vary between, let's say, solitary and pair living, you'd call it IVSO. And another species would show the variation between pair living and multi-male, multi-female groups. So do you categorize them the same way in these phylogenetic trees? And what do you think that means in terms of interpretation?
No, of course, you're right. These are different things and we categorize them differently. The only example I showed now here with the primates, it was one of the first analyses. We didn't have the statistical tools yet, but this is also what we are working on now, that because if you live in a pair and solitary is something different than if you live in multi-male, multi-female and one male, multi-female group. So this has to be differentiated, which is then coming to a question also of statistical power, and which is why we searching throughout the entire mammalian or the mammalian clade. And this is then also, of course, to come to a better understanding why we need data from more species in the future. But of course, it's something different. And I mean, in the primate example, we then differentiated it by having the different forms of IVSO where solitary living is involved to see whether this might explain our discrepancy to previous studies. And it was not the case, but it's important to be able to do that.
Okay, thank you. Also, follow-up to that is with some of the younger scientists in mind, what you see might be dependent on climatic factors at the time of the study or population density, sex ratios, things like that. So it sounds like you've said this before, I think in some of your published papers, that you'd need to study those species, those populations for a long period of time like you've done to get a really good idea of what their social organization is, right?
Yes, that's a lot of work. I mean, getting the database on all mammals and social organization is difficult, but getting these life history and ecological data in is not easier, especially like what Eduardo showed with Pantheria. I mean, this database might not be too reliable as well. And we would like to add climate data to see whether we can explain evolution of stability versus IVSO, but this is also a lot of work. And so there's still lots of things to be done. And yeah, of course, one needs to get these life history data, environmental data, but our point here is these data also have to be reliable, and people always have the aim to get as big database as possible and then include assumptions, don't include not so accurate data, and we try to avoid this. And this is first a lot of work to find the good data, and second, it is frustrating because often the data is not there. So like you said, everybody who is doing field studies should include such measures and factors as well.
There's no one else in the queue at the moment. Are there anyone else want to comment or have a question? Or yeah, David.
Hey, thanks for the great talk, Carsten. It was super interesting. I'm wondering if in your exploration of IVSO, if you've noticed that some forms of IVSO seem to be more common across multiple species or across multiple clades.
I don't know. My feeling is that when they're group living, there's more often IVSO between, let's say, one male, multi-female and multi-male, multi-female groups, but I don't know if that is really the case. So to be honest, I cannot answer this question, but it's interesting to look at this. What I can mention here is that one mechanism that can lead to IVSO is what we call social disruptors, environmental disruptors. This is if the social organization doesn't change because individuals change their tactic, but let's say there's a pair living species and the predator eats the male, then there's a female left. So this is something that's imposed from the outside. And we always try to find information on this in the papers if we can exclude such environmental disruptor events, and often we can. But this would also lead to the prediction that species that have as the most common form of social organization pair living should show more often IVSO in our database if there is a disruption by the environment, especially by predation. That is something we also want to test. And this is very important because we have to differentiate between cases where IVSO comes into existence because it is the result of evolved individual tactics versus when it's imposed by them on environmental disruptors, because this we cannot explain with evolutionary theory. This is something else.
So I have a question from a number, 782653.
Can you hear me? That's probably me. My name is Maren Hook.
Yes.
It was probably the way I logged in. Anyway, I obviously very strongly agree with your plea for more natural history data and the importance of studying not only one population, but many populations. Do you have any suggestions how we can convince funding agencies and university policies to fund this and to support this rather than just the so-called high impact studies that are based on the groundwork, but and the groundwork doesn't get funded, but the high impact ones get funded. The so-called high impact.
Yeah, it's similar to what somebody asked before. I don't have the good strategy, but it is clear to do good life history studies. You don't only have to invest money, but you have to invest a lot of hard work. So we also have to convince students. And like what I said before, if you were able to find funding for a field study on one of the great species, consider taking together with your PhD student, a master student who benefits from that the field vehicle, the field station is paid for this study on this great pair living species. Consider taking a master student along who has then the possibility to study one of the non-spectacular species. And I think this is how we have to start. We have to start from scratch with small projects. And then I'm sure we will find something interesting and fascinating and hopefully find the funding for the questions that arise from this. Like what Sulema said, we have to do descriptive studies before we have hypotheses, but this will lead to new hypotheses that hopefully other people will also find worthwhile funding. The other point, of course, can be our role as referees and maybe editors, that we also promote these studies, and we don't send back a paper and say, oh, this is not theory driven, it's only life history, but we say, wow, great, you spend so much time and energy in studying this strange animal, it's really worthwhile. So maybe this is one thing that we can do, but it's no easy avenue. You know how difficult it is already to find funding for these great species, and yeah, it's one of the most difficult things we have to do to find money.
Any other questions or comments? Yes, Nelson.
Hello, I hope you can hear me.
Yes.
Yes. Carsten, thank you very much for your presentation. I really enjoyed it. What I understood from your presentation was that all mammals are socials, but the degree has a huge difference between every single species, right? And also, I'm not sure if the right way to analyze all this data will be using categories. I don't know, what do you think about using numbers to do better comparisons, for example, using social networks to create a better categories? Or I don't know, what do you think about that?
Yes, of course, you're right. The easiest example I can find for this is not social organization, but mating system, where the question will not be, is the mating system monogamous or polygynous, but what is the degree of extra pair paternity, for example, where one can very easily really put it in numbers with the mating system. If it's from 100% to 50% with the social partner or even less, that would be easy. Then for social organization, it's the same, like I think what I replied to Peter Kappeler is that the first thing to do is to put an IVSO category threshold. If it's 10, 20, 30% of the social units that differ from the main form of social organization. Then how to put this in real numbers? I'm not a statistician, so I wouldn't know how to do it. But if somebody can do it, of course, it would be a great improvement. But I'm not the person who has the expertise for that. But if it's possible, it would be very good.
Okay, thank you.
Any other questions, comments? There's currently no one in the queue.
So maybe some questions still come up, but I would already like to say I enjoyed this evening a lot. I have to say, when we started it, I thought it's easy to be the first person talking, but today I was very nervous talking to so many people from all over the world, but especially as I knew my talk is a bit critical, but I really enjoyed the audience here. And I think it was really very nice for me. And I'm looking forward to all the other lectures we're going to have here. And I really would like to thank everyone who participated today. And I hope there will be even more people in the future. And there are many more interesting talks to come.
Yeah, I'll follow up. Thank you, Carsten. I really enjoyed that. And again, I enjoyed the interaction afterwards. The next week, there will be, I'll be giving a talk on the Daegu study. Trying to follow up Carsten here is going to be a big challenge. So I really enjoyed it, Carsten. Eduardo, do you have anything you want to add before we sign off?
Thank you, everyone. I also really enjoyed it. A special thanks to Carsten and Lauren. Apparently, Carsten led, Lauren followed, and they were very kind to invite me to join them. But it's been terrific couple of months. We've worked so hard. We thought so much. We prepared so much for having this wonderful first meeting. I thank you all, and thank you all for joining us. And well, let's hope to, like you said, I mean, it'll be a tough one to keep up with what Carsten did, but we'll try. So we'll see you all next Tuesday for Lauren's presentation now.